I think today's lecture will be the final one on enzymes. The parts of the course which is on enzymes, enzyme kinetics. I don't know. Okay, yeah, maybe. Yes, because with the enzyme kinetics, we'll also look at some applications of these enzyme kinetics in particularly the pharmaceutical industry, how this is used in drug development. So that after this, we can move on to the other part of the course, which is on biomembranes. All right, so we did look at enzyme mechanisms the last time, and so we continue today to look at enzyme kinetics, which is the aspect of enzymology that studies the relationship of the rate of reaction to the parameters which we looked at. If you remember, we did look at parameters that affect enzyme activity, looking at enzyme concentration, substrate concentration, pH, temperature. So when you do experimental works in the lab such that you study the rate of reactions depending on the different parameters. That is, if you vary those parameters over say some period of time, and see how the rate of reaction is related to these parameters, what you're doing is you are doing some kinetic studies. Indeed, the curves that we saw when we were looking at the factors that affect enzyme activity. So if you remember, we looked at the relationship of enzyme concentration on the rate of reaction, and we saw the curve, which was a linear curve, showing a direct proportionality between enzyme concentration and the rate of reaction. That curve is a kinetic curve. The same way when we also looked at the relationship of substrate concentration on the rate of reaction, we also saw the saturation curve, which showed that there was at the beginning a direct proportionality between the concentration of substrates with the rate of reaction, and then at a point when you had the system saturated with substrate, then you begin to have the curve flattening out and plateauing, becoming giving a constant rate of reaction. And then the relationship of temperature, pH, which also showed some optimum temperature or optimum pH, but below that there will be a decrease in the rate of reaction, and above it too, you saw also a decrease of the rate of the reaction. All those curves that we saw were all kinetic curves. So simply that approach, the study of the mechanism of enzyme activity depending on the rate of reaction. When you keep all conditions constant, but you vary only one of them, so that you see the relationship that the rate of the reaction has with the different values of those parameters, then you are doing some kinetic studies. Before we continue, let's take a look at some historical background of this aspect of enzymology. So the first representation of the relationship of say substrate concentration with the rate of reaction, which was expressed mathematically. First of it was by Victor Henry, who was Russian French. He was born to Russian parents, but was born in France, and so on paper he was French, but his parents were Russian, and so he studied in France, in Paris. He first had his PhD in psychology, and later went into chemistry. He did have a second PhD in chemistry, and did studies on enzyme mechanism. Using at the time the enzyme which was used by many of these chemists to study enzyme activity was invertase, sometimes being referred to as saccharase. It's the enzyme which is responsible for breaking down the disaccharide sucrose, the table sugar, into glucose and fructose. And so to study the rate of this reaction and come up with a mathematical representation of how the concentration of the enzyme or the concentration of the sugar was related mathematically to the rate of reaction. Now it got inspired from earlier chemists who worked on these things. A professor who was working in an industry, a professor in brewing and malting. And so he got this idea and came up with this equation, which could be used mathematically to represent the rate of a reaction based on the concentration of the substrate. But at the time, the scientists didn't fully understand the equation that he actually developed. It was after about, say, a decade that the German Leonard Michaelis, he was German, but he was a Jewish. His parents were Jewish, but born in Germany, and was working in the universities. At the time, Leonard himself, though he was interested in doing scientific research, scientific research wasn't something which fetched money. A lot of people, like scientists at the time, were not financially stable. And because of that, he decided to do medicine first. So Leonard was first a physician, medical doctor, before later he went into scientific research, chemistry. And together you had Maud Lenora Menton, who was a Canadian. She also was one of the first women who did medicine in Canada and wanted to go into scientific research, but also at a time in Canada, women were not allowed to go into scientific research, and so she decided to leave Canada and go to Germany. And when he got to Germany, he worked together with Leonard, and they came up with this equation, which was referred to as the Michaelis-Menten equation. And it is this Michaelis-Menten equation which has been used over the years to now, is the simplest form of representation of enzyme kinetics that you'll come across. There are others, but this is the universally form of Michaelis, the universal form of enzyme kinetics that is being used over the years. Currently, that's what is being used. Right. Now, we can take a look at what this Michaelis-Menten kinetics is. So the Michaelis-Menten kinetics, took into consideration factors such as the substrate concentration. You can see the curves which we saw much earlier. See that the concentration is on the vertical axis and time is on the horizontal axis. And they studied these factors such as substrate concentration over time, product concentration, so the product which was being formed is going to be the glucose and the fructose that were formed. And they also took into consideration the fact that you have enzyme that is in a reaction mixture, which is the enzyme which is going to catalyze the reaction. But they also took into consideration the fact that at a point there will be the formation of what is referred to as the enzyme substrate complex. So if you remember, we did look at the fact that every catalytic reaction. Okay, so somebody says my voice is very low. I don't know why, because my microphone is I checked the control. It's very high, so I don't know. Okay, maybe let me check the computer itself and see if maybe on the computer there is it's also high, even almost to the maximum. So I don't know why. Yeah. Maybe you should check the volume of your speaker and see. Perhaps the issue is there. Okay, so the microphone is so close to my mouth too. So, all right. So you had the enzymes. You will see after the lecture, when you listen to the video, you see how high the volume is. So I'm sure maybe something about maybe your speaker is what is causing it. All right. So you had these factors: substrate concentration, product concentration. I should change the output from phone to speaker. Okay. You are telling. Okay. Gabriel is advising Adiza to change to set your the output on your phone. If the phone is a phone you are using, you use speaker, not the phone. If it is phone, it will be so low. All right, so you have the substrate concentration, product concentration, enzyme concentration, and then the enzyme substrate complex, which you do remember we talked about the transition state of looking at the profile of every catalytic reaction, that the enzyme substrate complex will be the transition state, and it is from that transition state that you then have the formation of products. So they took all these factors into consideration, which when Victor Henry first developed his the first equation, did not take into consideration at time zero some of these factors of, say, the enzyme substrate complex. So this was a more comprehensive study of the enzyme kinetics, which became more clearer to scientists, and from then they could easily apply this to enzymatic studies. Now they noticed different regions of this reaction. So the first region was what was referred to as the pre-steady state, which was at time zero, where you have the substrate concentration being high. Because, of course, at the beginning of the reaction, you have the substrate in the reaction mixture that has not started yet changing into products. At the same time, you have the concentration of the product being low. So you see the curve for the substrate is black, and at time zero, that substrate's concentration is high. And at time zero, you see there's no product yet formed, and so you have the product concentration being zero. And you notice there's that steady rise of the concentration of the product over time, all the way until it gets to the end of the reaction where you have product concentration being high. And if you compare that to the substrate, when the substrate begins to get converted, the concentration begins to gets low and low until all the substrate is consumed in the reaction mixture, and so the concentration of the substrate becomes zero. Now, how about the enzyme? How is the enzyme going to fare in this reaction? Again, at the beginning of the reaction, you have the certain concentration of the enzyme. The enzyme is free at that moment. It's not bound to any substrate molecules yet. And so you have the enzyme concentration being high. That is, all the enzyme molecules were free. And compare that to the enzyme substrate complex. No enzyme substrate complex is yet formed at time zero. And so you see the enzyme substrate complex is also zero. But as the reaction begins to proceed, you have the enzyme substrate complex increasing all the way until it gets to a point where you have all the enzyme molecules getting saturated. And so when you have all the enzyme substrates, you have all the enzyme molecules getting occupied by substrate molecules, and you have this enzyme substrate complex, you have what is referred to as a steady state. Because you remember, it is at this point that you don't have any change of the rate of the reaction. No matter how amount of substrate is there in the reaction mixture, there's not going to be any difference in the rate of the reaction. And so in that region, you have what is referred to as the steady state of the reaction, where you have the concentration of the enzyme, and at the same time, the concentration of the enzyme substrate complex being constant, relatively constant, because as you see, the enzyme concentration lowers and becomes almost at the same level, constant throughout. At the same time, you have the enzyme substrate complex, because all of it is occupied. You have at that moment a steady state reaction, a steady state of the reaction. And then once you have the substrate being consumed and you having formation of products, you start having the enzyme substrate complex concentration also going down, because then you are having the substrate concentration almost showing that you are all the substrate molecules are getting consumed in the reaction, the process of the reaction. And so gradually you have the transition state, which is the enzyme substrate complex, reducing until you get to zero concentration. Whilst now you are having the enzymes getting freed, because once you are having product being formed and the substrate is getting consumed and all of it is almost not available again in the reaction mixture, then all the enzyme molecules are going to be free. And so now you have free enzymes at the end of the reaction. So at the end of the reaction, you have concentration of the product at its maximum. you have the concentration of the enzymes now equal to the concentration of the enzyme at the beginning of the reaction. Because as for the enzymes, they are just catalysts. They are not being converted. They only come in to increase the rate of the reaction. And at the end of the reaction, they are free and they will remain in the reaction mixture together with the product. Same way, you have the enzyme substrate complex. All of it also gets converted into the product. So what you have at the end in your reaction mixture will be the product and the enzyme. And so that was the kinetics that Michaelis Menten, were able to, explain and made it so clear to all scientists to understand the mechanism of reactions, pertaining to these parameters. And so it is actually out of these that we could have those curves for the various factors that affect enzyme activity, what we looked at in the last lecture. And so there was a mathematical representation, simplifying the relationship of the rate of reaction to substrate concentration. That you have V0, which is, say, the rate of the reaction, say an initial rate of reaction, is equal to the maximum rate of reaction, what is being written as Vmax, times the concentration of the substrate divided by the Km is what is referred to as the Michaelis Menten constant, and again plus the substrate concentration. So this value, this equation is generally used in enzymatic studies. Now, what is a Michaelis Menten constant? Now, looking at a curve which we saw before, which is the relationship of the substrate concentration against the rate of reaction, you realize there is a maximum rate of reaction. That is the rate at which you have that the maximum rate of reaction, which at which that rate becomes constant, where you have the curve flattening, where you have that saturation of the reaction mixture and you have the curve flattening, becomes the maximum rate of reaction, because you cannot go beyond that. Beyond that is not possible. So you have that maximum rate of reaction. Now, which is here, you see it being estimated with that dash line. Now, then what is the Michaelis Menten constant? The Michaelis Menten constant is expressed as half of the maximum rate of reaction. So you see that is being shown there that the Michaelis Menten constant is the substrate concentration at which half of the maximum rate of reaction is reached. So again, the Michaelis Menten constant is the substrate concentration at which half of the maximum rate of reaction is reached. And so you see these constants being used in enzymatic, in calculations of enzymatic reactions. I said there are other forms of the kinetic representations of enzyme activity, what's being referred to as the Lineweaver-Berg, Eddie-Hofstee, and some others. But this is the simplest, and this is what is generally used. And so we'll focus on this and do this at our level. The others, their estimation too are not so accurate compared to this. So this is the most accurate, where actually this equation, if you just take a tangent of of this curve, and you remember the equation of a straight line, you represented in the form of y equals ax plus b, where you have that y being the rate of reaction and a being the slope or the gradient of that tangent which you have taken of this curve, and then a b being the intersection of the line with the y-axis as an equation of the line. You can derive this equation from there once you know that you have these values such as the half of the maximum rate of reaction and Michaelis Menten constant, which is Km. So which means that the slope is going to be giving you the that's Km, over the a half of the maximum rate of reaction over Km. I mean, you can derive these things and when you work it out, you come to reach this. But there's no point actually going through all of that. Once you have, because this equation is already well known, is there and is being used all the time to study enzyme kinetics. Okay, so that is it about the Michaelis Menten equation. Now we can look at enzyme inhibition. I think inhibition and inhibitor were terms that we came across when we were looking at terminologies in enzymology. And we did talk about the fact that inhibitors were substances that either slow down or they prevent reactions from occurring. Those are inhibitors, as opposite of activators that we did refer to as some of these activators were the coenzymes and the cofactors which we looked at. Now these inhibitors mostly are applied as pharmaceutical agents. I mean, that is to inhibit a particular reaction from occurring. Because once the reaction mechanism is known, it's known that there is an enzyme that catalyzes a particular reaction. If a substance is known to be an inhibitor of such a reaction, then that substance, which is the inhibitor, could be used to regulate that reaction. We'll see some specific examples of the use of these inhibitors in drug development. And then, of course, how, generally how these enzymatic reactions, knowing the mechanism of these enzymatic reactions, can actually be applied in different ways. Okay, now inhibition itself, we can do some classification of inhibition itself, so that we have what is referred to as competitive inhibition as one of the forms of inhibition that we'll see. And in this case, you do remember that an enzyme is having a particular part of it which is referred to as the active site, where you have the catalysis of the chemical reaction occurring. Particularly, it is at that active site that you have the substrate binding. So this is being shown as if this is the enzyme, the red is the enzyme, and the groove in it is the active site. Then you have a normal substrate that comes into that active site, gets bound onto the enzyme, and there is that reaction on the substrates, and the substrate can be converted into a product. So you have that interaction between the enzyme and the substrate. How about if you have an analog say a structural analog, a compound that has a structure just like the substrate of that enzyme. But it is not the substrate, but it looks like it. And it can, because it's a structural analog, and remember I said a structural analog, it's not a functional analog, meaning that that substance is not having the same function as the substrate, but it looks, structurally. That is, if you look at its chemical structure, it looks just like the substrate and can bind into the active site of the enzyme. It will bind, but it will not result into a chemical reaction, which means it only binds there and prevents the enzyme from binding onto its substrate, so that once it's not able to bind to its substrate, the chemical reaction, which is supposed to be catalyzed by that enzyme, will be prevented. So that is competitive inhibition. Competitive because the substrate and the inhibitor are competing for the active site of the enzyme. So that is why we refer to that as competitive inhibition. Now, competitive inhibition can be shown with the Michaelis-Menten plot, what we just saw. We can use that to represent the how competitive inhibition will look like. So looking at this curve, you have the red curve for the normal reaction, that is without an inhibitor. You have the reaction proceeding along the red curve. Now with an inhibitor in the reaction mixture, which means you have the inhibitor binding onto the enzyme, what you do notice is that you have a shift in the Michaelis-Menten constant to the right. What does it mean? Now, in competitive inhibition, the maximum rate of reaction is not changed. It can still be reached. It can still be reached because if you have an inhibitor which is bound to the enzyme, is bound to the active site of the enzyme, but that binding into the active site is not permanent, it's not something which cannot be undone. You can add, when you add more of substrate into the reaction mixture, because you have increased the concentration of substrate, the substrate will go to displace the inhibitor from the enzyme, so that in the end, you can still reach the maximum rate of reaction when you increase concentration of the substrate. And so if the maximum rate of the reaction is constant, then of course, the half of it is also constant, is unchanged. And so you realize that Vmax over 2, which is the half of the maximum rate of the reaction, is the same for the normal reaction and the inhibited reaction. So that is for competitive inhibition. But the maximum rate of reaction will be reached at a higher concentration of the substrate than for the normal reaction without inhibitor. And so similarly, you have the Michaelis-Menten constant, which is the concentration of the substrate at which half of the maximum rate of reaction is also increased. So that is why you see that there is that shift of the Michaelis-Menten constant to the right. So practically, when you are doing this in the lab and you have a reaction mixture, let's say you have some sugar and you have some amylase also in the reaction mixture with the sugar, so that you have an amylase that will be acting on the sugar. And you introduce an inhibitor of amylase that goes to bind to amylase so that the reaction is slowed down. Now, in order for you to increase the rate of the reaction, even in that condition, all you needed to do is to add more sugar into the reaction mixture. Once you add more sugar into the reaction mixture, that sugar will go to displace the inhibitor from the amylase, and the amylase is going to be free now to carry out the reactions, to catalyze the reactions, the reaction on the sugar. And so you increase the rate of the reaction, even though the inhibitor is there, because you now have more substrate. The substrate, goes to, it's competition. So the more molecules of sugar you have, the sugar will win that competition. That's what it means. It means the probability of the enzyme binding to the substrate, you have increased it by increasing the concentration of the substrate. So the sugar wins the competition. All right, so that is to put it in simple English language for us to understand. Okay, how about if you're having an inhibitor that binds to not the active site of the enzyme, but you have some other site of the enzyme, what is usually referred to as some allosteric site of the enzyme that you have an inhibitor binding to. So the inhibitor doesn't need to bind to the active site of the enzyme. And so what you see is you have this the green structure, which is representing the enzyme. The active site is there, and you have the normal substrate that is bound in the active site. But you see there is that site, the allosteric site on the enzyme, that you have an inhibitor that can bind onto it. That we do refer to as a non-competitive inhibition. It's a non-competitive inhibition because the substrate and the inhibitor are not competing for the active site of the enzyme. And so you see that even though you have the substrate binding into the active site of the enzyme, once you have the inhibitor binding to that allosteric site of the enzyme, it prevents the reaction from proceeding. I mean, this is being depicted as once you have the inhibitor binding to the enzyme, it changes conformation of the active site so that the active site is not fitting to the substrate, so that there will not be any activity on that substrate. And thus the reaction is inhibited. All right, now how is this going to look like by the Michaelis-Menten plot? Now here again, we're looking at the Michaelis-Menten plot where the red curve represents the uninhibited reaction and the blue curve represents the inhibited reaction. But this time around, it is non-competitive inhibition. Now, what do we notice? We notice that there is a change in the maximum rate of reaction. Yes, there is a change in the maximum rate of reaction. The maximum rate of reaction is lowered. It's lowered because this time around, it's not a competition for the active site between the inhibitor and the substrate. So whether you increase the concentration of the substrate or not, that will have no effect on the inhibitor, because the inhibitor has its own site that it is binding to on the enzyme. So it will still have that maximum effect of inhibiting the reaction, so that once you have the maximum rate of reaction lowered in non-competitive inhibition, you also have half of the rate of reaction also being lowered, even though you have the Michaelis-Menten constant being the same. So the Michaelis-Menten constant is not changed. Remember, the Michaelis-Menten constant is that concentration at which half of the maximum rate of reaction is reached. So the same concentration of the substrate at which half of the maximum rate of reaction is reached for the uninhibited reaction, that same concentration gives you half of the rate of the half of the maximum rate of reaction for the non-competitive inhibition, except that it is a lower value for the inhibited reaction. All right, so you can compare these two, and then you can see the difference between competitive inhibition and non-competitive inhibition. Right. Now you can also have what we refer to as reversible and irreversible inhibition. So you have, so as the word suggests, reversible. Reversible means that the binding of an inhibitor to an enzyme can be undone. I mean generally, medications are reversible inhibitors. Medications that are designed with this principle of going into inhibit chemical reactions are reversible inhibitors. They bind, so the drug, the medication will bind to an enzyme, but it's not going to be permanent. It can get unbound after its activity. And there's always this sort of an equilibrium between the bound protein, that is the bound enzyme with the substrate, the bound protein with the inhibitor, and the unbound. You always have that. And then you also have what is referred to as the covalent inhibitors, which will be the irreversible, because covalent bonds, once that is like a very strong bond between the inhibitor and its target protein, which is the enzyme. And once it binds, it becomes like a new compound which is formed altogether, a complex that cannot be undone. This you see in situations of maybe toxic substances that can bind onto proteins to form some maybe protein adducts, or which can result into maybe binding to some enzymes like acetylcholine esterase, which will be resulting into maybe some paralysis. Let's say in the case of pesticides, for instance, that are used, to control pests, they go to cause some paralytic effects in these organisms. We'll see some of these examples. We'll see them clearly. So you can have what is also referred to as reversible and irreversible inhibition. So we've looked at 4 forms of inhibition already. First, we look at competitive inhibition, looked at non-competitive inhibition, and now we are looking at reversible and irreversible inhibition. Okay, now let's begin to look at some specific examples, because what we've just talked about have just been general things, but in order for us to see specifically how these things work, we need to look at some specific examples so that we can have a deeper understanding of this. Right, so the first that we'll look at is say the control of the process of biosynthesis of tetrahydrofolate. And quickly, where did you meet tetrahydrofolate? Tetrahydrofolate was one of the coenzymes we talked about. We refer to it as folic acid. You do remember that? Folic acid. All right. And folic acid, we also mentioned that folic acid is that coenzyme which is involved in many reactions of cell growth and cell proliferation, and we talked of it as being needed, required for, say, in human development, for instance, when a fetus is developing, the folic acid is required, because at that moment there is a vigorous reproduction of cells, and also specialization of the cells, what we refer to as cell proliferation, which is why the lack of it can result into particularly abnormalities in neural tube formation, as like abnormalities in the brain formation. That is the reason why a lot of times for pregnant women, they are prescribed this folic acid in order to prevent, things like mental retardation in babies. Now, similarly, folic acid is also required by bacterial cells, the same way that these human cells require folic acid to grow, to proliferate. The same way bacterial cells also make use of folic acid. Now, when we look at a process of the synthesis of folic acid, so if we take it as in bacteria, the bacteria makes use of, it's the same process which occurs in humans as well. We also do synthesize folic acid, except that if it's not being synthesized in adequate amounts, that's when supplements are needed, like the folic acid tablets that are prescribed. Now, the process of biosynthesis of the tetrahydrofolate, from top coming down, these are just some 3 reactions. You have aminobenzoic acid, which is blue, which is getting conjugated with the dihydroterine pyrophosphate, which is the red molecule. So the blue molecule, which is the aminobenzoic acid, sometimes also being referred to as para-aminobenzoic acid. These two combine. And we also refer to the fact that a pyrophosphate is a diphosphate. It's a form of an activated form of the dihydroterine. And so you have that molecule activated by those 2 phosphate groups. Just look at it as it's going to be a conjugation, or say a joining between these 2 molecules, the blue molecule and the red molecule. And that is exactly what you see below. The product that you see there, the dihydroteric acid, is just a conjugation. It's a joining of the red molecule, the blue molecule, the para-aminobenzoic acid, and the dihydroterran pyrophosphate bind to give you dihydroteric acid. Right. The dihydroteric acid gets converted into dihydrofolic acid by conjugation of, look at that, there's a green portion that has come to bind onto that blue portion. That is glutamic acid. That's an amino acid that comes to bind on the carboxylic acid group. So that's a peptide-like bond. That is actually because you don't have the para-aminobenzoic acid is not an amino acid, so we can't say it's a peptide bond per se, but we can say it's a peptide-like bond because it's just like a peptide bond which is formed between a carboxylic acid group and an amine group without glutamic acid or glutamate. So you have a dihydrofolic acid, which further now gets converted to the tetrahydrofolic acid. What we well, one of the forms of the folic acids that's you find the folic acids present and active in the body or even in bacteria that are using these folic acids in their metabolic reactions or in their metabolic processes. And so, but when you look at this, the dihydrofolic acid and the tetrahydrofolic acid itself, I mean, what's the difference? You can see that if you take the red part of the molecule, there is no hydrogen on that nitrogen there, and there is a double bond. I mean, looking at the two red rings that are fused together, but when you come down, you realize now there is a hydrogen on that nitrogen, and the double bond is also removed. So that's a reduction reaction there, and it's an acceptance of a proton. One of these reactions which we looked at when we were looking at the classification of enzymes, that is addition of groups that removes double bonds from molecules, and at the same time also is a form of a reaction. So you have a reduction of the dihydrofolic acid to tetrahydrofolic acid. It's just an observation of, say, if you take dihydrofolic acid and you take tetrahydrofolic acid, you try to look at a molecule and see what is the difference between them. And you see, oh, there's no hydrogen here and there's hydrogen here. There's double bond there, there's no double bond there. That's all, just to notice those differences. And that's all. It's nothing complex about it. But there are some small notes that I want to bring your attention to. For instance, look at this. The very first arrow where you have the reactants, the para-aminobenzoic acid and dihydroterran pyrophosphate reacting, and you have that arrow going to the next, you see on that arrow there is a cross, and beside it, it is written sulfonamides. And sulfonamides are a group of antibiotics. So what it means is that those antibiotics inhibit the enzyme dihydroterate synthase. It is that enzyme which catalyzes that first reaction. And there is another note here, the third arrow, which is also showing the conversion of dihydrofolic acid to tetrahydrofolic acid. There too, you have an antibiotic, trimethoprim. So trimethoprim also inhibits the activity of dihydrofolate reductase, which is the enzyme responsible for the conversion of dihydrofolic acid to tetrahydrofolic acid. Now let's take a much closer look at this, taking the reactions one by one. So that first reaction, and then the third reaction where you have those notes, where you have the sulfonamides acting. Now so remember, the enzyme which is to be inhibited is dihydroterate synthetase. Now, the substrate to that enzyme is the para-aminobenzoic acid, which is also referred to as the 4-aminobenzoic acid. Now look at this. You see sulfonamides, so one of these sulfonamides, or a common sulfonamide, which is sulfanyl amide. Say one of the simplest of these sulfonamides, sulfanyl amide. If you take a look at it, the structure, and look at the structure of the para-aminobenzoic acid, which is a substrate to the enzyme, you see there's a close similarity of the structures. The only thing is that for the sulfonamides, you are having this sulfoamine group in place of the carboxylic acid group, which you find in the para-aminobenzoic acid. And so the sulfonamides are competitive inhibitors to the para-aminobenzoic acid for the enzyme dihydroterate synthetase. So you see what I was saying, that you see that the structures look alike. And so sulfonamides are structural analogs of the substrate para-aminobenzoic acid, so that when you have sulfonamide binding to the active site of the enzyme dihydroterate synthetase, then it's like the enzyme is blocked, the para-aminobenzoic acid cannot bind to the active site, and so the reaction will not be catalyzed, the reaction will not proceed, so that you have the synthesis of the tetrahydrofolic acid occurring. And once you don't have synthesis of tetrahydrofolic acid occurring, then of course that is going to disrupt the growth of the bacterial cells. It's not going to result into the death of bacterial cells, but it's going to inhibit growth. So the sulfonamides are what you do refer to as bacteriostatic substances. If it was going to result into the cell death, we will say it's a bactericidal. We'll come to see some bactericidal activity also as well. Okay, so I see Deborah has written something on WhatsApp that most people are unable to join the class. I mean, what's the reason? I don't know what it is, because everything has just been as it has been before. You are 77 participants here now. All right. So that is an example of competitive inhibition, which is being applied for this group of antibiotics, which are sulfonamides. Okay. So now we take a look at that third reaction also, that we saw that note there, trimethoprim, which is also an antibiotic that is also used in maybe the treatment of some urinary tract infections and some other conditions. And you see also that trimethoprim is an inhibitor to the enzyme dihydrofolate reductase, which is the enzyme responsible for the conversion of dihydrofolic acid to tetrahydrofolic acid. Now, if you take a look at trimethoprim and you take a look at, say, the substrate, which is the dihydrofolic acid, not so much of, I mean, there's some similarity, but the similarity is not so close as you see for what we saw earlier for the sulfonamides. I mean, if you look at these, the rings, the two rings here, you see there is no fusing. There's no fusing of this ring to that, except that, yes, you have some similarity of the very first ring there to the first ring of the dihydrofolic acid. And so this brings you to what we do talk of as specificity of these substances. So that there are some substances that are more specific than others. So we do say some of these antibiotics are, say, broad spectrum. So they act on a large group of bacteria. I mean, and at the same time, the side effects of say some of these medications, because they are not all that specific. They have more side effects acting on some other systems than only what they are intended for, and that you have much more of side effects of these substances, because you can see clearly that trimethoprim, which is supposed to be, say, a structural analog to dihydrofolic acid, competing for the active site of dihydrofolate reductase, is not very much alike to that of the dihydrofolic acid. Even though, yes, it acts as an inhibitor, it binds to the enzyme and has that effect of inhibiting the enzymatic activity of dihydrofolate reductase. You see that the similarity in structure is low. All right. So that is, looking at some antibiotics. Now, that same reaction that we just saw, we also have the medication methotrexate, which is also used for, some in chemotherapy. And that is to control the growth of cancer cells, because of course cancer cells, their proliferation, their growth is also going to follow, the use of folic acid. If you remember, that folic acid was in nucleic acid synthesis, DNA. And so if you have the nucleic material from cancer cells being, reproduced, then there will be more reproduction of these cancer cells. But methotrexate comes to inhibit, the synthesis of the tetrahydrofolic acid, which acts as a coenzyme in that process. Now, let's take a look at the structure of methotrexate. So if you look at the structure of methotrexate here, you notice that, yes, this is much, much, much more alike, just like the dihydrofolic acid, the substrate itself. You can see the two fused rings there. The only thing is, in place of the hydrogen, which is on that nitrogen, on dihydrofolic acid, you have in methotrexate a methyl group, a CH3. And also methotrexate has an NH2, an amine group on that first ring there. In place of it, you have oxygen, cyclic ketone group in dihydrofolic acid. And of course, you can see also a different position of this double bond. There's a double bond here in methotrexate. Here, the double bond is rather with the nitrogen up there. So there are these slight differences, but apart from that, I think all the other aspects of the molecule are the same. Particularly the glutamic acid portion is the same, bound to that ring there, the six-sided ring. So everything else is the same apart from the differences which we just pointed out. Okay, maybe you can also see that there's a double bond with this nitrogen in the second ring, and but this here there is rather hydrogen on the two nitrogens. We have on the two nitrogens of the fused rings, you have hydrogen on them, but in methotrexate you don't have any hydrogen on those nitrogen. They rather have double bonds on them. So you have these differences in the molecule, but you can see that methotrexate is having a much more similarity than trimethoprim, what we just looked at, which means methotrexate will be more specific, much specific to the inhibition of these dihydrofolate reductase. And of course, that should tell you that this is really much more needed, particularly also because if you know that in chemotherapy, so if you're using methotrexate in chemotherapy, for instance, and you see we are talking about inhibiting the biosynthesis of tetrahydrofolic acid. And we're saying that even normal cells require the tetrahydrofolic acid, to grow, to reproduce. And which is why you have in chemotherapy, you do have this effect of maybe scaling of the skin, hair loss, and all of that, of fibrility of the nails, because even though the medication is being taken to control the proliferation of cancer cells, they can also go to affect normal cells as well, in addition. And so you have some of these side effects there. But of course, it's much more specific than trimethoprim, as we have just seen. All right, so these are some specific examples. Now let's move on to another kind of antibiotics, which this time around they are bactericidal. They will result into the cell death of the bacteria. And so if you take, first of all, let's take a look at, say, the general structure of, say, a bacterium. So if maybe this, we're just taking some bacillus, that's the very dark, the thick black portion of that bacterial cell is a cell wall. And that cell wall is made up of what we actually saw earlier when we were looking at mechanism of action. We saw lysozyme, which was responsible for breaking down, this molecule that is the major component of the cell wall of bacteria, You remember? Peptidoglycan. So here you are seeing it, the peptidoglycan layer. So if, the thin black part of that bacteria which is being shown it has a plasma membrane, the cell membrane. We'll come to also look at the plasma membrane much more, which is a bilipid layer. And covering it is the cell wall of the bacteria, which is made up of peptidoglycan. Now, what is this peptidoglycan? So the peptidoglycan is it's a polysaccharide which is made up of. We did see this the structure in the last lecture as that structure is a derivative of monosaccharides and acetylglucosamine and N-acetylmuramic acid. Here they are represented by these violet and blue circles that alternate. So you have N-acetylglucosamine binding to N-acetylglucosamine, and then the next is N-acetylmuramic acid, like that. So you have that alternation. Then you also see that there is a linkage between these, oligo. Let's it's an oligosaccharide. Derivatives of these monosaccharides that are binding together, and they are cross-linked by some peptide. So you have a two tetrapeptide chains which are cross-links holding these oligosaccharides together, and that structure is what we are referring to as peptidoglycan. I mean, from the name you can tell peptido means there is some peptide in there. Glycan is carbohydrate, so it tells you that you have peptides and some carbohydrates, these are some oligosaccharides which are coming together to give you that structure, which is giving you the layer of the cell wall, giving protection to the cell membrane of the bacteria. All right, that understood, what we do refer to as gram-positive bacteria. You've done this in microbiology, I think. If not, maybe you are doing it now, or you do it. Now, we want to take a look at the action of a common antibiotic, of the class penicillin. How does penicillin, affect the growth, or even resulting in the death of bacteria, these gram-positive bacteria? And to do that, we look at what we refer to as the synthesis of the peptidoglycan biosynthesis, because it is that synthesis in which you have an enzyme which is involved in that synthesis of the peptidoglycan that the penicillin targets. So that penicillin goes to inhibit that enzyme to prevent the synthesis of the peptidoglycan. So once synthesis of peptidoglycan is inhibited, there won't be cell wall for the bacteria. The bacteria cannot synthesize its cell wall, and once there's no cell wall, the bacteria is not protected, and it will result into its death. Now, the enzyme which is being targeted by penicillin, because it is that enzyme which is being targeted by penicillin, you see that it is a lot of times referred to as penicillin binding protein. But it's also referred to as a glycopeptide transpeptidase. That is if we're referring to its function. It is an enzyme that catalyzes, the transfer of these peptides that will result into bonding between them, have these cross linkages between them. Then that is how you have the peptidoglycan synthesized. But if you're looking at that protein in terms of the fact that it is the protein, or it is the enzyme which is being targeted by penicillin, then it's being referred to as penicillin binding protein. Now that penicillin binding protein is having an active center in which you have what? A reactive serine. You do remember when we were looking at the mechanism of action of enzymes, one of the mechanisms we looked at was the serine proteases, where you have in the active center of this enzyme serine, which was responsible for donating proton. So there's a proton transfer from serine onto, say, an imidazole ring, which activates the enzyme. That mechanism, you can go back to it and look at it. So here too, what we are referring to as the glycopeptide transpeptidase, or the penicillin binding protein, is a serine protease. Now, what happens in this process of peptidoglycan biosynthesis? Here again, we have some keys here. You have the red. These red spheres represent alanine. The blue spheres represent glutamic acid, and the green spheres represent lysine. I mean, this is just to show that it is these amino acids that bind together to form the tetrapeptide on the glycan, this N-acetylglucosamine, N-acetylmuramic acid, which will then bind. You have these tetrapeptides, which are on the N-acetylmuramic acid residue. So the N-acetylmuramic residue is also these green portions of the oligosaccharide. And you have the teal color, which is the N-acetyl the N-acetylglucosamine. So you can see that if you look at number 1, you have N-acetylglucosamine portion. The next green is the N-acetylmuramic acid portion. And so you see that alternation throughout. And the tetrapeptide is on the N-acetylmuramic acid residue throughout. You see that tetrapeptides, which consist of alanine, the red spheres, green, the lysine, blue, the glutamic acid. So you see that. And then you have this, say, a pentaglycine chain. So there you have this glycine, amino acid glycine, 5 of them, in having that chain there, the smallest of the amino acids having that chain on the lysine residue. So you have the penicillin binding protein, or that glycopeptide transpeptidase. What is it doing? It is catalyzing the linkage of that pentaglycine chain onto, the terminal alanine on another tetrapeptide, which is on the N-acetylmuramic acid residue. Because in the end, if you look at number 5, what you should have is this linkage. You have that yellow linkage, binding between, the green sphere and the red sphere. So that's going to happen for all of these. Green sphere, red sphere, green sphere, red sphere will be binding together and have such a cross linkage, so that you now have 2 chains bound together as one, two chains as one, two chains. And those two can also, like another one can bind on top, like you have all of these bound together and you have that full structure of the peptidoglycan. And so that whole process is being catalyzed by this glycopeptide transpeptidase. All right, so now, knowing that it is this glycopeptide transpeptidase that catalyzes, the formation of these cross linkages, which results into, the synthesis of the peptidoglycan. Penicillin is then used to inhibit that enzyme, which is why it's being referred to as the penicillin binding protein. So in this case, this is an irreversible competitive inhibition. And the enzyme we have seen already to be the glycopeptide transpeptidase, which is to be inhibited. The substrate in this case, we say it is that alanine, the bond which is formed there. You have if you look at the structure of the penicillin binding protein, there is one short end. In the active side, it looks like a chair. There is one short side and there is one long side. And then you have that connection between them, which is where you have the formation of the cross-linkage occurring. And when you pay attention to the tetrapeptides, you realize that the ends of these tetrapeptides are alanine. So because at the end here, it's ending with two red spheres, which are alanine residues. That's why you see that the substrate is being referred to as D-alanine peptidoglycan ends, because the peptidoglycans, they end with the two alanine residues, and they fit into those sides, the long side, the short side, before you have the cross linkage that will be formed between the two. That's what you see in number 2. You see one of the long chains, almost all of it gets into that, the long sides, and the shorter side you have the other alanine binding in there, and you have that cross linkage forming between the two of them. So that bond formation there is being catalyzed by the glycopeptide transpeptidase, or the penicillin binding protein. And remember, it is a serine protease. It's a serine protease. So that the active site is serine. Now, what do you see? We've brought here penicillin, these structures which are looking like 9 or 6. These are penicillin molecules. And you see these penicillin molecules, they are close. But interestingly, just in the presence of the enzyme, the penicillin molecule opens up and fits into the active site of the enzyme. And once it fits into the active site, it blocks the enzyme. And once the enzyme is blocked, it cannot carry out that biosynthetic reaction. There will not be any formation of these peptidoglycan, the peptide bonds, that's the cross linkages which are supposed to be formed in that biosynthetic process. And that is how penicillin inhibits the growth of the cell wall of these gram-positive bacteria. Now, structurally, if you look at the chemical structure of penicillin, you see penicillin is having that 2-ring structure. You have this 4-ring. What is being referred to as beta-lactam ring. You hear of these a lot in Ghana. I think some of these companies that produce these pharmaceutical companies like maybe Entrance Pharmaceutical, Tobinco, and others. They do produce some of these, you know, penicillin-based antibiotics, and they do refer a lot of, to this beta-lactam. Is this a group of antibiotics? They are all in the penicillin family. And if you compare that to the substrates, the substrate that you have this penicillin competing with, what you notice? You notice that that's the peptide, the alanine terminus, which fits into the active site of that glycopeptide transpeptidase, is only like it's an open ring. If you look at this, it's much more of looking at the penicillin, which is opened. The ring is opened. So which is why, first of all, you see that the penicillin, yes, is closed, but then it opens when it reacts with that serine active center. So that's why, penicillin sometimes, it's also referred to as some seal side inhibitor. Like the enzyme, it sort of, it deceives the enzyme. The enzyme thinks of it as if it is its substrate. Then when it binds into it, it results into the enzyme not being active any longer. So yes, the structure, if you look closely at it, looks much just like that, the substrates, the alanine peptidoglycan ends, which bind in the active site, except that if you look at the penicillin molecule itself, they have close rings, but when it comes into contact with the enzyme, the rings open up and the structure looks just like that, the alanine peptidoglycan ends. All right. And now, this is just to show you much more. So if you have a glycopeptide transpeptidase, which is having a serine residue, which is just represented by, say, there's a hydroxyl group in the side chain of the serine residue, which is in the active site of the enzyme. And on that serine residue, you have penicillin binding, and when penicillin binds on that serine, you now have this complex between the penicillin and the enzyme formed, and that renders the enzyme inactive. So that complex is what is being referred to as the penicillyl enzyme complex. And so you have an inhibition of that enzyme there. And so you see, I was talking of the fact that you hear a lot of these pharmaceutical companies talk about beta-lactam antibiotics, because all these antibiotics have that same active ring, the ring that opens up for the penicillin molecule to fit into the active site of that penicillin binding protein. They look alike. And so if you look, you have different, forms of these beta-lactam rings from left to right. You have what is being referred to as a pinam, and then you have piname. So the difference between pinam and piname is just, there's a double bond in piname here, which is not found in pinam. You also have carbapenem, and the next will be CFAM. So that CFAM is having now this sulfur here, and this ring is now a six-sided ring. if you look the first 3 rings, you have a 4, like a square-sided ring with a 5-sided ring fused to it. You see the fourth one is now a 6-sided ring. That's a C-farm. And then if you now have a double bond in there, you now have a C-film. I mean, that's not all. You have some other rings anyway. It's not exhaustive, but generally these are the rings that you see. So, for instance, a common antibiotic like amoxicillin. What is the ring in there? I think that ring is the first one. It's a P-NAM ring, which is in amoxicillin. And you have this combination of amoxicillin and clavulanic acid. On the pharmaceutical market in Ghana, you have amoxiclav, which, because you do have, clavulanic acid and amoxicillin, the combination, I mean, deals with antibiotic resistance, so that you have a different ring in clavulanic acid, which is not even represented up here. This is an oxypenam. It's just that, in place of the sulfur in the penam ring, you have an oxygen there. You have an oxygen there. So you do come across this when you go. I mean, those of you maybe going to do some industrial attachment in the pharmaceutical industries, or even later working there, you come across something like this, beta-lactam antibiotics, and you'll talk about the beta-lactam ring. So if you hear the beta-lactam ring, this is what is being talked about. Now we do also have what we refer to as prostaglandins, which are like some natural analgesics in the body. I mean, these are products of the metabolism of this fatty acid, arachidonic acid, which results into these different forms of prostaglandins, prostacyclin, thromboxanes. I said they are like natural analgesics because thromboxanes, for instance, will have the effect of blood clotting. I mean, it's a way of controlling the amount of blood that flows to areas of inflammation. Prostacyclin will do the opposite of thromboxanes, so you have this regulation of inflammation. So the pain which is felt during inflammation is reduced by these prostaglandins. Now, I mean, knowing this, I mean, let's pay attention to some things. For instance, you have this cyclooxygenase 1 and cyclooxygenase 2, and these enzymes, are catalyzing this process of the conversion of arachidonic acid to these prostaglandins. I mean, so we can focus on this cyclooxygenase, which is being written as COX-1 and COX-2. Now, what do you see? We do have some painkillers, some analgesics, what we refer to as, this, for instance, brufen. And brufen is an inhibitor of cyclooxygenases. So this enzyme, the COX-1, COX-2, which are responsible for that, metabolic reaction of arachidonic acid to prostaglandin. So in that manner, the brufen is able to, control the recognition of pain in inflammation. All right. Similarly, you have, say, aspirin. Aspirin Aspirin is also an inhibitor of this. Cyclooxygenase 1, again, that same reaction where you have arachidonic acid being converted to prostaglandin. Aspirin comes to inhibit that. And once aspirin inhibits it, of course, there's going to be that reduction of recognition of pain in the end. So that's how these painkillers also act. I think this should be the last one, where you have what we refer to as diisopropyl fluorophosphate. This is something which you find in insecticides. It's like an active ingredient in insecticides, and this acts on the enzyme acetylcholine esterase. Acetylcholine esterase, even for us humans, is responsible for muscle contraction, muscle relaxation and contraction, so movement of the muscles. And the substrate for acetylcholine esterase is acetylcholine that acts as a neurotransmitter. It sends signals to the muscles, the muscle cells, to contract and relax. So for instance, if you have acetylcholine binding onto an enzyme, binding onto the muscle cell, it causes, contraction. Then acetylcholine esterase comes to hydrolyze the acetylcholine from the muscle cells, and that will result into relaxation of the muscles. So if you have an inhibition of acetylcholine esterase, what then happens is that process of contraction relaxation will not be going on, and that results into a paralytic effect. So the insecticide usually is a nerve agent that goes to cause paralysis in the insect. So the insect is not able to move. So some of these substances like, these DRFP, chronic exposure to some of these, even in humans, can result into serious problems, health problems. Because the accumulation of these over time, for instance, many of these substances are lipophilic, and they can get accumulated in the adipose tissue. And maybe in small amounts, you may not feel the effects immediately, but when they get accumulated in the adipose tissue over time, you may it's not seen that it's there, but later when perhaps there is some disease condition, some sickness or old age, that one begins to lose weight, then these substances which are stored in the adipose tissue begin to be released into the bloodstream, and that is when they start to have serious effects, when they cause some of these paralytic effects. Once they inhibit acetylcholine esterase, it can result into paralysis. That is bad for the nervous system, which is why it's a serious thing to consider the indiscriminate use of pesticides, especially by farmers that have low or none of education about the use of chemicals, and indiscriminately spraying these chemicals all over the place. Sometimes they get themselves exposed, and even others get exposed to it, and later results into serious problems. So you see that the active site of an enzyme, that is the acetylcholine esterase, is also a serine protease. So the active site serine is having that formation of a covalent bond with that phosphate group of the dipropyl fluorophosphate, so that in the end you have this complex being formed between the enzyme and DRFP, which renders the enzyme inactive. All right, so this is where I will stop talking, and then I open the floor and now listen to you. If you have any questions, if you have any contributions, you have anything to say, the floor is open. You can speak. You can raise your hand. I will enable your microphone, call your name, you speak, I respond to you. Or you can write in the chat and then I will respond to it. The floor is open. All right. I see Emmanuel Dacha, your microphone is allowed. You can unmute yourself, speak.
Hello, sir. Okay. So I wanted to ask, because I've heard stories of people saying that, let's say you have used an insecticide spray on a piece of land for a very long time. It will reach a point where it will no more affect the insects that it usually kills there. Is it that they get evolved or they get used to the insecticide spray, or what? Or what really happens there?
Okay, so that is some form of resistance that some of these organisms develop over time. So it's what happens is that when you are using some of these chemicals on the land as you have put it, what is seen is that some of the insects die, but some of them that do not die when the insecticide is applied to them now begin to get adapted. It's like there is an inducement of some mutation in them, and they begin to develop some resistance to that particular chemical, which is actually bad because then in the end, the insecticide will no longer be able to do the work it's supposed to do. Because once you are having such mutations occurring in these organisms, they will begin to reproduce such strains of these resistant organisms over a period of time. So that is what happens in such a case.
Thank you, sir.
All right. Okay, we still have some time. I think about 16 minutes more. If you have any questions, you have any thing you want to say, you can put up your hand and then I will call your name. I will unmute your microphone, then you speak. Or alternatively, you can write in the chat and I will respond.